by my office. You know where it is? On the third floor, and there's a cubby hole right next to the elevator. Ask anybody.
Should I come with the rest of the
No. Yeah, if you need some of these questions, ideal thing that I can do back to the community. Batch.
Yeah, we're not here, but it's okay.
Otherwise, I'm feeling like a shoe job, another one, like 4 times the same thing.
Yeah.
One person, one time. Okay. Okay. Huh? Yeah, I think so. Because of the mic, I can't move around. Right? So, otherwise, I was going to say, can you? Yeah. You can. Okay. Well, what do you want? What's best for the client? What do you like? You like that one better?
Yeah,
Okay. Okay. Yeah, thank you for introducing us. We should probably be going. I'll make some introductory remarks while people are coming in. Just I list the objective usually, if I remember. And then regarding textbook, it doesn't matter to me what textbook you want to read. Whatever you feel comfortable with. Some people like certain textbooks, okay. But for the exam, etc., most of the assessment will come from the PowerPoint presentation. Okay. My recommendation is to try to understand at least some memorization, vocabulary, for example. There's nothing you can do about that. I'll try to introduce them as gently as possible. But if you can try to understand the concept of what's going on, you're much better off. That's it. Welcome to the physiology part. And some people are tempted to take notes. That's okay, but you'll find yourself losing track of the slide presentation while you're writing. And then 2 slides down, you're asking me to go back 2 slides. You missed something. Okay. So I have provided you with notes. Okay. Whenever it's, you can take a look. It's on Moodle, but there's notes at the bottom. Some people, and also all the I post everything in the PowerPoint. For example, if this is too small, you can go in there and change the size. Not a problem. Same thing with any writing. I tried to. Can you read this okay? Okay, then we should be okay. Live Rio. Okay. The digestive system can be, we're going to be covering it in a different way than most textbooks do. Some, like most textbooks, for example, will deal with sections on motor activity, absorption, secretion.
The mic.
Do what now?
Closer to the mic, please.
Now?
Yes.
Better? Okay. And if I have a tendency to go too fast, so if I'm going fast, just try to slow me down. All right, we're going to follow a bolus of food from ingestion to elimination and deal with different organ systems that the food bolus either interacts with secretion, okay? And this is from another Tim Barrett's textbook on GI, and I have access to it, but it's stolen from somebody else. And basically, this is the digestive system as a machine. Okay, you have a chopper, you know what those are, right? Mastication. You'll have a blender, acid sterilizer, okay, served as a reservoir. And then you have a reaction vessel, which would be your intestine, into which you put in a detergent, the liver, enzyme, and bicarbonate from the pancreas, okay, neutralize gas. And then you have this catalytic cycle, that's where all your hydrolysis will occur within the small intestine, pretty much 90%. And then you have a residual combustor, like that one, desiccated, remove the final water and create a pellet, okay, in which you expel feces, okay. We control things on the way in. Okay. On the way, you can't see the top part. How do you control? You control things on the way in as an adult. As a newborn, you don't. But as an adult, you control things on the way in. You put the food in your mouth, or whatever you decide to put in your mouth. And you can control the output. Okay. You have control over that. Everything else that occurs in there, you have no control over. All right? It's all run pretty much by the intrinsic nervous system, autonomic, hormone, but you don't even know it. Right now, you could be processing, kind of late for breakfast, but you could be processing some food, and you don't know what's going on. You have no idea. You're paying attention to me. Some of you are anyway. Okay. So you have some control on the way in and on the way out. Also, it's good thing there's terminology here. Describe the process of movement of fluid. And this is a cute little thing. The GI lumen. Think of it as inside your body. It's really outside. Okay? That's what this slide sort of illustrates. This is outside your body, inside, outside your body. Your lungs, right? Outside air. That's the same thing in the GI tract. Okay. And so when we talk about exchange of fluid between the blood, this is the orange part here, cardiovascular system, blood vessels, you can talk about absorption in terms of something moving from the lumen of the GI tract into the blood vessels. These are supposed to be red blood cells. Okay, that's the blood vessel. Or you can talk about secretion, and to secrete fluid outside of your body into the GI tract, it has to come from somewhere. Guess where it comes? The blood. Okay. So sometimes that terminology is flux, blood to lumen flux, or lumen to blood flux. Okay, that's all we're talking about. If you can remember that lumen is outside your body, that's one step ahead. How do you control this food? Once you ingested the food over here, and it's going to go through your GI, how does it do that? Well, you haven't had much anatomy yet with GI, right? But you're going to have muscular layers, okay, surrounding the GI, all the way, probably to the end. You're going to have muscle. And this down here shows you this is supposed to be a bolus of food, and motor activity is going to move it down, basically, down through, and all these reactions will take place. What regulates that, we'll cover a little bit today, is the mesenteric plexus. That covers the motor activity. And that's located between the circular muscle and the longitudinal muscle. If you haven't had that yet, we'll go over it again. But anyway, there's some food now. Food is what you take in. Sometimes people refer to a food bolus when you're swallowing it. Once it starts mixing with digestive secretion, it's called chyme. Gotta memorize that term again. It's called time, that's vocabulary, and you can do about that. And then at some point in the large intestine, and colon, it becomes feces, right? Yeah, regulation. We introduce you to enteroendocrine cell. Again, vocabulary is not that hard. Entero, anytime you see enteric, entero, that's intestinal, right? That's what that word means. And endocrine, you have what hormones are, things like that. No? Yes? You don't even know what a hormone is? Okay. Well, let's take a look at that then. Enteroendocrine cell. The old textbooks use other terminology, but there are some diffuse neuroendocrine cells or something like that. But they're not, they don't come from the neural crest. They come from stem cells. We'll cover that. Stem cells in the crypt. They don't come from the neural crest. So the GI tract generates these cells itself. And if it's a hormone, then it's going to release something called secretion. And these, you have a circulation, you have to put the portal vein pretty much drain the entire GI tract. And then return the blood to the heart, pump back. Okay. So this secretion enters the bloodstream, goes to a distant site, and exerts its effect. Okay. And that's over here on the target cell, all right? That's hormonal. And a paracrine effect is when the same cell can release something and it goes right next door. It doesn't have to enter the circulation. It's a diffusion right next to another cell type and exerts its effect. Okay, that's called paracrine. Para means next to, right? Okay. All right, an example of hormones, you'll see that your intestine here are going to send signals to your liver for the detergent supply, those are going to be bile salts, or to the pancreas, those are distant organs. Adjacent targets can be epithelial cells or nerve. Let's take a look at that. This is the general concept of enteroendocrine cell in the GI tract. Okay. There's different types. Open type. All that means is that it has processes that are open to the lumen, to the outside of the body, and they're sensitive. Okay. It can sense what's out there. Good, bad, etc. Some people call them taste receptors, but you know, in the intestine, I'm not sure you taste it, but nonetheless, they're sensitive. And you have closed type, which are not connected to the lumen at all. So the only way you can activate these cells is from the blood side. Okay, that makes sense. This is the lumen outside, and this will be blood. Here's the capillary. Okay, let's take a look at A, B, C, D. A, paracrine, I mentioned it before. Some cells, these cells, these are in the notes, what I'm saying, so don't worry about that. They might have processes that are coming in close proximity to another cell. This is paracrine, and affects this particular cell, which is a gastrin releasing cell, the G cell. And then you can have a paracrine cell. Here's another one. This is paracrine as well. But this one now communicates to nerve inside the interstitium. In the interstitium, you'll have blood vessels and nerve, and then some immune cells and things of that nature. Are we okay so far? I'm going too fast, let me know. Okay, and then the closed type, of course, you have to come from the circulation, like gastrin will be coming from here, go into the bloodstream, go all the way around, come back, and this is going to be your endochromatin-like cell. You'll get this stuff in histology. You have a question already? Slide number 4 or 5. What's the question? I'm sorry, I don't know.
The D, G, and myself.
The D is paraclint. Oh, the D? Outside this has processed in the, this will be like your eye cell. There it is. This is going to release cholecystokinin, for example, in response to lipid. It's going to activate nerves right there in the interstitium. The interstitium, we said blood to lumen. That's easy to remember. But along with the blood vessels are nerve in that same compartment, interstitium. Okay? Is that okay? Yeah. We'll see that. As a matter of fact, just try to understand what's going on, get the big picture here. You want to memorize it? Probably not. I'll tell you what, you can use this entire presentation today as a review if you want. You'll know this by the time you get done. You don't have to memorize it. You'll understand what's going on. Okay? That's the important part. What's interesting here is we have in bold, gastric, CCK, CPK, and then remotely. What do you want me to do? That's what they're called, right? Anyway, but you'll see, you'll hear this so often that it shouldn't be memorization at all. Okay? And the cell types that make it and what they do. Okay? These 4 in bold are the only known. They have all the key characteristics to be called hormones. Okay, they enter the blood and can affect target tissue at a distant site at the right concentration. Okay, ones with the asterisk are paracrines. Okay, so like 5-hydroxytryptamine or serotonin here, it operates next door. It's a paracrine. It doesn't work by going. It's not a hormone. It doesn't enter the bloodstream. Those are just triggered by asterisk. And in the red, it's going to be one of your favorite peptides, somatostatin. It's the only one that inhibits it. Okay. That's all you have to remember. And it'll serve you well. And when you get into endocrinology, I don't know what that is. Next year. But you hear somatostatin, make sure it's inhibited. You have to take a guess at something, it's going to be inhibited. Okay. And you know, it's just the only one. All the others do something in a positive manner. They increase. Okay? You don't have to know. Let's go over this. Because these peptides can be endocrine, paracrine, we don't generally classify them into families based on function, but more on structure, activity, relationship, structure. Okay? Amino acid composition. Tertiary structure in some cases. And you can, these families, secretin family, and the ones that are secreted in response to a meal are the gastrin and secretin family. And then there's also peptides that are released between meals when you're fasting. Okay. And let's take a look at the secreted response to meal to gastrin and secretin. So I'll never use this term again. It's called pharmacophore. It's defined in the notes again. All that means is structure required for activity. They could have just said that, right? But that's what they call it. So it's the structure necessary for activity. And in this case, what they're outlining here, this is gastrin. And you know that gastrin 17. Gastrin actually circulates in 2 forms, the large form called big gastrin. It doesn't matter, we're not going to get into that. But the important part is outlined here in red. The last 4 amino acids here are identical to the structure of another member of the family, cholecystokinin. Okay. And here it's CCK8 because it's 8 amino acids, gastrin 17, over 7 to add 9. But there's an overlap because, and notice that this is gastrin, but it'll interact with CCK2 receptor. It won't interact very well with a CCK1 because it doesn't have a complete pharmacology. But they have some overlapping function. Here, the terminal amino acids are identical. Yeah. Want me to do it again? Well, I have a hard time hearing you guys in this auditorium. Screen. Which part? Pharmacophore? It's just a terminology, it just means the structure. You know what amino acids are? Okay, this is the peptide. Okay, this family, called the gastrin family, includes gastrin and CCK. Okay, these two, gastrin and cholecystokinin. The last four amino acids are identical. Okay, so some of their activity may overlap, and they're put in the same family. And all you need is the last 4 or 5 amino acids. As a matter of fact, clinically, they use pentagastrin, which is only the last 5 amino acids. You can buy it. Okay, so you don't need the whole gastrin molecule. All you need is the terminal end. Okay, that's required for activity. That's why we're showing you just the terminal end of oxalate. Okay, it's in the notes as well. The secretin family has nothing to do with the terminal amino acid. The homologies are internal. You can see in the blue, the same amino acid. Now here, you cannot use truncated form. You need the entire molecule because it's the tertiary structure that's important, not the terminal end. So clinically or otherwise, we have to use the whole molecule, otherwise it's not going to work because it won't configure itself at the tertiary structure that interacts with its receptor. You've had this, right? Tertiary structure. Okay. The two members of the family that we'll be dealing with is VIP and secretin. And again, they'll have secretin as both endocrine therapy function. VIP is a neurotransmitter, but it'll also induce secretion and stuff like that, and also be endocrine therapy. Okay, so the entire peptide is necessary for activity. We'll cover ghrelin and motilin. Ghrelin comes from the stomach when you're hungry, and motilin comes from the intestine to clean it out between meals. And we'll cover those functions later. We're just covering some concepts now. This is enzyme secretion. I think you know that. In terms of signaling, the only thing pretty much that I'm going to be interested in is going to be calcium and cyclic AMP. Have you had those before? Amino cyclic AMP? Yes, okay. And that's how it works. And the other thing here you have acetylcholine. It's obviously a neurotransmitter. You had that already, but last. And it increases calcium, and this would be an acinar cell in the pancreas, makes enzyme. And what happens is calcium stimulates enzyme secretion. Now, secretin and VIP in the same family, secretin will increase cyclic AMP, but it's not a major stimulant of enzyme secretion. Acetylcholine and cholecystokinin are. Now, by having two pathways, you actually potentiate. So if you have acetylcholine and then you co-stimulate with secretin, you obviously are increasing enzyme secretion even more and co-potentiation, okay? Because there's two different intracellular signaling pathways involved. That's as complex as you want to get. So they can potentiate each other. I don't have the time, so make sure let me know when. Otherwise, if it's finished, take the rest of it. We're supposed to meet again or not. Okay, let's do nerves. These are called neurocrines, if you will, like VIP or something like that. And here we're doing, obviously, we're an action potential, and it works on a target. So let's take a look at some of this. Now, I mentioned in the introductory slide the myenteric plexus. This is the nerve plexus ganglion cell between the longitudinal muscle and the circular muscle. Longitudinal muscle is going to be on the outside. Circular muscle, the major player that we're going to be dealing with, is going to be on the inside. And this myenteric plexus regulates motor activity. Okay, this is what's going to help you move through mixed food. Okay, the myenteric. There's also a submucosal plexus, and it's located in the submucosa right below the mucosa. And guess what it's going to do? Most is going to be involved in secretion. Okay, things like that. But let's take a look at the myenteric plexus. Now, in the title, the little brain of the gut. Okay, as I mentioned before, you're not going to know what's going on in the GI tract at all. Once you swallow the food, that's it. You know, you don't know what's going on after that until you have the urge to eliminate them. Okay, then you'll wake up again. But at any rate, the GI tract, we say that the amount of neurons present in these plexi in the GI tract is equivalent to the spinal cord. Those sides of the person counts all these things. But anyway, it's about 1 million of them. And, but the point being is that you can cut all the autonomic nerves to the gastrointestinal. It doesn't alter its function. Okay. Motor activity. It just goes down. Autonomic nerves help facilitate, but it doesn't need, you know, you can take it all, cut all the nerves out, and the GI tract is going to process from here. Okay. That's why they call it a little brain, kind of brain of its own. And that's a helpful thing to remember when we're dealing with things that happen in the GI tract. That the lumen's outside the body, and that the GI tract has a brain. Okay, in a sense, primitive but a brain. These are motor patterns we're going to cross. So, we still okay for time? We could push through if you want, but just let me know. Okay, there's peristalsis, and that's going to be propulsive over here. Moves material, okay, distance. And here are the sites where you'll find this kind of activity. The esophagus, when you swallow, okay, it's going to move the food into the stomach. The stomach, in turn, is going to empty into the intestine, and the intestine is going to move it along to the colon. Okay. And then you have the mixing that occurs primarily in small and large intestine. And that mixing, it does alternating constriction, relaxing. That's all we're doing. No, you know, it's mixing. And then you'll have tonic. You'll have tonic contraction. That's sphincters that block passage, separate compartments. We'll get into that. But maybe you will. Yeah, that's good. You don't have to be in class. They have to be in class. No, I disabled that. Just save your time.
Wait outside. Outside. Wait outside.
You will be outside.
Yeah, yeah. I'm outside.
See, this is what happened. I can't touch it.
So you'll have to deal with him outside, okay? You'll have to deal with Wild Dahan outside. All right. Hopefully, there's not too many people that had a problem. There's always a problem.
The Wi-Fi was so.
Look, you have, I don't know anything about them. You have to talk to Wild Dahan outside. That's why we don't do it here. You want to listen to the lecture, you can stay here. You want to talk to Wild Dahan, you have to go out there. I don't have any control over it. Sorry, I'm not trying to be rude, but in the next 20 minutes, you'll see the line over there. Okay. All right, we covered it, okay? There's going to be 3 types of movement. We'll deal with this. You'll understand it. And you can see that there's nodes. Okay? These are going to be the neurotransmitters we're going to be dealing with. And again, if you want to memorize it, go ahead. But you don't have to. You can understand it by the time it's done. And as I mentioned earlier, this whole presentation, basic concept. To review too, when you're done and you're studying for the final, you want to go back to the basic concept. All this should come right back to you immediately. If it doesn't, find out what's going on in the PowerPoint presentation. But you mentioned VIP. Some of you, they relax smooth muscle, okay? Some contract, acetylcholine, muscle. But you'll know that now that you know acetylcholine contracts, you're always going to know that. You'll learn that gastrin releasing peptide. It stimulates, guess what? Gastrin releasing. Like some of these names are okay. Oh, GR. Nitric oxide, I don't know what that is. Smooth muscle relaxant. Vasodilator. We'll come back to this. Throughout the course. This is my taking apart the autonomic nervous system. The entire picture. I have been intended at the end, but it would have been too small. You wouldn't have been able to see anything. But you can go through that, or you can simply have whited out certain things, black certain things out, do whatever you want. But the whole picture at the end. And this is dealing with the parasympathetic, okay? Cranial output. The vagus is going to be a major player in the GI tract. Okay? Vagus, cranial nerve 10. And it's involved in the upper, and you can see it. Now, in terms of the pancreas and liver, the preganglionic fibers, they're shown here in a solid line. Preganglionic. And the little black things in here are intraparenchymal ganglia, liver, and pancreas. And here, in the GI tract, lower part of the esophagus, stomach, and proximal colon, the parasympathetic vagus is going to come down and interact with the little brain of the gut, the entire nerve. How that works. Okay. Anyway, the vagus will come down, preganglionic, and postganglionic. Is the entire nervous system, the little brain, that's for postganglionic, okay, in the GI. The sympathetic, they're going to come, heavy covered with also pelvic nerve coming lower part of the colon. Get to that. The sympathetic innervation, And here you have the preganglionic fibers, again, the solid. They go to the celiac ganglion. You have it, right? And the postganglionic fiber directly impact on the organ, or the postganglionic fiber go to the enteric nervous system. Okay. Let's see how that works. This is the cartridge. I took it right because I modified it. Some problems with it. And what I've done is I've focused on the mind plexus and to screw in my smooth muscle here. Talking about motor activity. And basically, remember we said we have an endocrine cell. They're a sensory, okay? They respond to chemical chemoreceptors. They respond to mechanical deformation, the mechanoreceptor. That terminology, you have to memorize, you already know what it means, right? The chemoreceptor can be chemical. Mechanical, mechanical. So they're going to respond to something. Take there, nerve, and I have the 1 and 2 cover that. But I want to point out, we mentioned that we covered autonomic in terms of vagus. 80% of the fibers in the vagus are there. They're coming from the GI tract. This is something they're sensing. 80%. Then ether, as I mentioned before, the sympathetics is the postganglionic, interact with the enteric nervous system, ENS. ANS stands for autonomic nervous system. Okay. The parasympathetic is the preganglionic interact, and then you're going to have some effect. Now, instead of memorizing something like this, let's just take it apart to what it looks like. Okay, let's take a look at smooth muscle. Now, I think we had Hushing talk about smooth muscle contract regulate. Let's take a look at that. And this is a cartoon. And here you can see the membrane potential. And you see it's not stable, like we have in skeletal muscle or striatal muscle, but it fluctuates, okay? Fluctuating membrane potential. And this is referred to as facial electrical rhythm. So it's a rhythm, and you'll find that different parts of the GI tract, intestinal colon, have different rhythms. This is going to be important to its function, okay? And PER just simply refers to how many of these cycles per minute. Just for example, if it's 1 minute, the flow wave, that would be the stomach. That's the characteristic PER of the stomach, 3 times per minute. And these slow waves can occur unless they reach threshold, no action potential, and no contractions. Okay, that makes sense? Now, when they reach threshold, you have action potential, development, or contraction of the muscle. Okay? The more action potential, the stronger the contraction. So pretty straightforward. And that's what I have here is threshold. Once you reach threshold, you have spikes with action potential. Okay. You have contraction. Increase the number of spikes, increase the strength of the contraction. Now, the problem lies with muscle. They took them out of the GI tract, fecal tissue, that they didn't have fluctuating membrane. They didn't even no slow. So now they're blaming all this on the interstitial skeletal Kahal. Kahal, I think he was Portuguese scientist or something, to describe himself and sort of name them. You'll run across this, and people name things that. So anyway, these cells are interspersed along with the enteric nervous system. They're mixed in, my enteric plexus, and the interstitial cells of the heart. You take these cells out, they have a vasoelectric rhythm. So these cells are considered a pacemaker. Now you're going to get that cardiovascular. Most textbooks you cover cardiovascular first, so you know what a pacemaker is. But most people know what a pacemaker is, right? It sets the pace. Okay. So these interstitial cells of the valve interact with the nervous system to set the pace. These are longitudinal fibers, minor flex, okay? So let's take a look at this now. Sympathetics come down on the parasympathetic. Now you can cut the sympathetic nerve and the parasympathetic, cut all of it, and the entire nervous system alone will run the whole genome motor. You don't need the autonomic, but the autonomic nervous system facilitates or inhibits the entire nervous system. Okay, kind of regulates it. Take a look at that. This is on the previous slide we looked at, and if you can look at it, I think it's large enough to see it. At any rate, these are the way it works. You're resting, let's say, slowly, there's pressure. Now, if you stretch the muscle, increase motor activity, you're going to do depolarize. So all these things that cause contraction, they're going to partially depolarize. So it doesn't take that much from the entire nervous system to contract. Okay. Now the opposite, neurotransmitters that are inhibitors, we'll cover this as well. Inhibitory neurotransmitters, they're going to hyperpolarize. So it makes it more difficult for the entire nervous system to generate contraction. This makes sense. Okay. And basically, that's all it's doing. Norepinephrine. VIP, vasoactive intestinal. It's a neurotransmitter, but it was first discovered as a vasodilator, increasing blood flow, and that's what it's all about. And NO, polarize. Make sense so far? Okay. All right, now I mentioned this, copy this, right? You'll have the nerves coming down right into these cells. Look at that. Now that's that doesn't happen, right? You have epithelial cell lining, and the nerves are in the interstitium. So I have to redraw it so that the nerve, okay, they don't communicate with the outside of the body. You don't have nerves coming out outside. But the nerves are in close contact with the cell. Remember the enteroendocrine cell, okay, your chemo and receptor, and they'll signal to these nerves. These nerves don't come from the epithelium. So they will trigger chemical or mechanical deformation depending. And you'll have short reflexes. Reflexes will come through here. And as I mentioned, you only know about it. Okay. We'll cover it. How do these enteroendocrine cells communicate? Right? We covered that. Now this is a villus. You don't even know what a villus is. Call it, this would be the epithelial lining of the small intestine. Projection. And here are your enteroendocrine cells, and it's responding to some chemical deformation. And it's going to release something, and right there locally, it doesn't have to go to the circulation. It's going to activate an intrinsic intrinsic to the GI tract. It stays within the GI. Extrinsic means it's going to leave the GI tract. Okay, if you are working for the vagus power, all right. So they can sense and enter on, they can tell nutrients. I said that some people refer to it as taste receptors. Those sense irritation. And the little brain comes in, how do you deal with stuff like that? And bacterial toxins, etc. And that's the way it works. Okay. Now we know how it works. We have to have the nerve, the interstitial. All right. Now, we have short reflexes. One, just go right through here. Has nothing to do. Has nothing to do with spinal cord, brain, in other words, peristalsis, overactivity of the okay. You can cut the autonomic nerve, they're going to have peristalsis. Not a problem. It's an intrinsic reason. Located within the GI, not dependent on any neural input whatsoever. But the autonomics can enhance it or diminish it. Okay, short reflex, not a problem. Long reflexes. And I mentioned short reflexes like peristaltic. Defecation reflex are also going to have a short component. It's also going to have a long component. Now, you can memorize these things like gastrocolic, but not into memorization. The first part of the word deals with the source. The second part of the word deals with the target. Okay, so something called the gastrocolic reflex coming from the stomach going to the colon. Does that make sense? All right. Enterogastric, enterointestinal, going to the stomach. Colon-ileal, that's what is coming from the colon. Now, do you have to memorize that? You see this and you're going to exactly know what's going on. That's important. Now, I have gastrocolic in red and ileocolic in red because they're going to be moving things down. Gastro, the other two reflexes in the middle, these two are in blue, they're inhibitory. So if you can remember this, it'll help almost 90% of the time. If something's coming from a distal part, interacting proximally, it's going to be slowing things down. If it's going the other way, it's going to be speeding things up. So any reflexes that are feeding back, they're going to be inhibitory. Any reflexes that are feeding forward are going to be stimulatory. Okay? Very cool. I think it's in the notes. Now, the long reflexes, some of these cover them now. Vagal. That's a common one that deals with different terminology used by different people, but basically it means that afferent or the sensing was in the vagus, and the efferent was also in the vagus. In other words, the vagus sensed it, and the vagus stimulated, like market, whatever it is. But the vagal is vague, it doesn't give you organ specificity. Also, these long reflexes that they end up going to your brain. Then you're going to be aware of your GI system. It's called pain. In other words, if something severe enough to become aware of it, it's a long, it started with Bobby, right? Kind of let you know there's something going on. These are long. Okay, and we'll get to this zipplication reflex with Bobby's spinal cord. Let me know about the time. We're okay, we can finish, and we come back after lunch too. So if you want to go straight through, 20 more minutes, we're done. You want to take a break? All right. Let me know when. The signal. Already? 1 minute? What do you want me to do in 1 minute? We'll come back.